diesen wunderbaren Wissenschaftler, den wir dank der Zusammenarbeit mit dem italienischen Generalkonsulat hier in Frankfurt, und da bedanke ich mich natürlich bei dem Generalkonsul Massimo Darchini, der Vizekonsulin Silvia Bianchi, die auch mit uns hier da ist, und dem Verantwortlichen des Kulturprogramms, Michele Santoriello. Wir arbeiten gerne und immer wieder zusammen, denn es gibt uns die Möglichkeit, italienische und deutsche Brücken zwischen den Kulturen zu bauen und hier auch interessante und einmalige Wissenschaftler, Wissenschaftlerinnen und Künstlerinnen einzuladen. At this point, I hope you know that we are speaking in English today, because Giorgio is holding his lecture in English, and therefore I'm switching right now. I take also the occasion to greet and welcome our colleague from Weltenleser, who is here. You know her as being one of the famous bookstores in Frankfurt, and she's here to introduce you and to offer you some of the publications of Giorgio Vallortigara. So, how we would like to introduce Giorgio's work is through asking how does knowledge and information emerge? What differences exist in the learning behavior of small children as well as young animals? Are the ways humans and animals perceive and experience the world perhaps not so different after all? Für Sie, die Sie ein bisschen mehr unser Programm kennen, Sie wissen, dass wir oft in diesen Interspezies-Fragen und Projekten auch mit unseren Kunstausstellungen unterwegs sind. Grund, weswegen wir Giorgio Vallortigara gerne heute zu Gast haben. The mind, which is your main argument, and argues Giorgio Vallortigara, is not a tabula rasa. Learning is only possible if a nervous system is already equipped with an inherent structure. Research on chicks, and we will hear some more about this in a minute, confirms the theory of the innate knowledge. This way of earning knowledge is not exclusive to us human species, and we share behavioral patterns, dispositions, emotions, and neural structures with other companion species, some of them separated from us by something like 300 million years of evolutionary terms. Just like human infants, the Kant's chicks, this is the title also of Giorgio's book, immediately look for their mothers. Giorgio Vallortigara weaves together ethology, and I had to look this up, what exactly it meant, and it's the study of human behavior and social organization from a biological perspective. That helps a bit more to decipher it. Neurosciences and a love for literature. And this is how I fell in love with all of your theses, through a wonderful book that unfortunately is not translated yet to German, but we will expect that. During today's lecture, he might share some surprising insights about the empathy of ants or the altruism of parrots, about the mysterious area of the brains where memories are stored to the biological origins, or the human belief in supernatural beings. Giorgio Vallortigara is professor of neurosciences and director of the Animal Cognition and Neuroscience Laboratory of the Center for Mind-Brain Sciences at the University of Trento in Italy. He's an internationally acclaimed speaker and scholar who has published the following books in Italian with Adelphi Editori, whom we thank also for this evening. Cervelli che contano. I took the liberty to translate this, although there is no translation available. Brains that matter. Pensieri della mosca con la testa storta, a wonderful book. Thoughts of the fly with the crooked head. And Il pulcino di Kant, Kant chicks. And the most recent book, A spasso con il cane luna, Walking the Moon Dog. Please welcome Giorgio Vallortigara.
Thank you very much, Franziska, for this kind and too generous introduction. and thank you very much for the invitation to the director, Franziska Nori, and to Michele Santoriello. I'm really very glad and very honored to be here. So I would like to start with a personal recollection. I spent most of my scientific career investigating the brain and the mind, the cognition of this little chap that I am showing you in the original form, namely soon after hatching, when it has still a little bit disgusting appearance because it's completely wet, so it's not fluffy and nice as chicks usually we are prepared to expect. And the question and the reason why I'm showing the newly hatched chicks is that perhaps you can wonder why. I mean, why a scientist should make use of chicks as an animal model. Of course, people interested in other things in life who are not scientists, maybe they are not interested in the particular reason why a scientist selects a particular animal model. But in this case, I think it is important to stress this aspect because we all have stereotypes about typical animal models in science, in biology. So mice, rats, maybe fruit fly for those of you who are more sophisticated, but why chicks? Well, one important thing is that if you ask what is the reason, the motivation, the rationale behind the choice of a particular animal model, particularly in the case of the mind, I think that people would expect that the main reason would be associated with phylogenetic distance, that is how much an animal is similar to us. And in this case, one would expect that in order to investigate the brain, the mind, the most plausible choice would be monkeys or anthropomorphic apes or things like that. But this is wrong because the choice of an animal model in science is not based on how farther apart is from us, from the evolutionary point of view, but on the very nature of the problem you are investigating. This is quite apparent in the case of fruit fly. You know that genetists, at least molecular genetists, use fruit fly, the Drosophila melanogaster, as a model. And obviously, this is an animal which is very different from us. It's an insect. But wrong or right, genetists believe that it could be a good model to study genetics because it offers some advantages. You can have progeny very rapidly. You can have giant salivary glands in the chromosome, in the salivary glands, and so on. In the case, and the idea underlying the use in the animal model is well expressed by a statement by Hans Krebs, a famous biochemist. Yes, it's the Krebs of the Krebs cycle. Some of you remember probably the suffering that has produced when we were at the high school. And he said, God had provided just the right organism with which to study any particular biological problem, which looks very weird. I mean, the idea that creatures are here in the earth just for the satisfaction of biologists is a joke, but it is also a serious joke in the sense that depending on the problem you are interested in, you select a particular model. So in my case, I'm interested in the origins of knowledge. What is in the head, in the mind, at the start, in a condition that using the Latin words you can refer to as statu nascenti. Is the mind a tabula rasa at the start, there is nothing, or is there something which is already written by evolutionary history? And in order to provide an answer to this question, we have some problems because on one side we would need an organism which is without any experience. So for instance, a creature which is studied just after birth. But at the same time, we need a creature which is also able to behave, to do something in such a way that we can test them psychologically, behaviorally. And this is something which is difficult to obtain in the classical altricial species, as we say, like rats, kittens, or even human newborns, because these organisms are extremely immature after birth, and therefore they do nothing, or there is a very limited behavioral repertoire which is available. But luckily enough, as Krebs suggested, God has made available to biologists precocial species. There are several, not just avian, but also mammalian species. For instance, young chicks or ducklings, guinea pigs among mammals. Guinea pigs and mammals are not very good models because they depend mostly on olfactory information, whereas we prefer to use avian models because they depend on vision, similar to human. And this is the reason why we use young chicks as a model. You see the point? Because you can say, well, what is the problem? Take a kitten or a young rat and deprive it for a couple of weeks of any kind of stimulus and then start your behavioral test after a couple of weeks. But this is not a good model, because obviously if you deprive an altricial species of the proper sensory experience for a few days or a few weeks, this would be not longer a physiological model, it would be a pathological condition. But in the chicks instead we can have an organism which is soon after hatching completely devoid of any experience, so you can check for possibility of experiences, including experiences in the egg, and at the same time it could behave in a very sophisticated way immediately after hatching. So I would tell you some stories about the idea of the innate mechanisms in the brain that have a sort of moral, a sort of message to take home. The first one has to do with magic. I don't know whether you are familiar with the famous trick of the amputated finger. This one, if you don't know it. Now you will see a version by a professional magician, and it is astonishing. Now this is a very interesting phenomenon from the perceptual and cognitive point of view. I don't know whether you never ponder about that, but given that the objects in our world are mostly opaque and not transparent, depending on a particular point of view, it is extremely rare that we can see objects in their wholeness completely. So for instance, in this moment, you can see me completely, but you don't have the impression that I am amputated at this level. Rather, you have the perceptual impression that I am continuing behind and beyond the occluder. And this phenomenon is so prominent that can be illustrated easily in this cartoon. Let's look at this image. There is a young girl partly covered by a fence. If now I open the fence, what you would expect? You would expect something like that, right? And you would be very surprised if I show you something like that. But in fact, from a strictly physical point of view, it is in terms of the rays of light, quantum of light coming in from the different part of the object. These are not reaching in any way your receptor, your eye. So this part, which are mentally present, are just a reconstruction of your brain, are not there. And the effect is so prominent that you could see a classical example, a bizarre one, a weird one, in Giotto, in the Cappella degli Scrovegni. Here there is the problem of the representation of the halo, and obviously, typically, this is represented as a complete circle, which is partly occluded by the head. And this is fine, but if Giotto has the problem to represent the saints from the back, and obviously he maintained the convention, and the impression in this case is that there is a sort of black disc occluding and preventing any vision from the apostles and the saints, which is bizarre. The alternative is to see something like a sort of U-shaped structure. You can say, well, what is the origin of this mechanism, which is called technically a modal completion? A modal because obviously there is no a modality producing the effect because there are no physical stimulation from the occluded parts. And the obvious explanation seems to be is the result of learning. We have experiences with objects in the world, and as a result of this experience, we learn to recognize them even when they are occluded. But several years ago, the Belgian psychologist Albert Michot argued that this could be wrong. Look at this image and try to memorize exactly the shape and to represent the real, the actual shape. Then cover it in this way with a finger. What do you see? I guess you see a triangle, a canonical triangle. And you can try to repeat the trick several times. Look at the image, learn about that. Stay looking at the image even a month, 2 years, it doesn't matter. Every time you cover it, you will see the canonical triangle. And this is not, Michot argued, due to the fact that we have familiarity with the triangle, because you can use even completely different objects, different from triangle. What is crucial is the collinearity that you could observe in these boundaries here. The brain has mechanism for interpolation, and so it is something like that. There is a boundary which is oriented in this way. Then there is an interruption, and then another boundary which has the same, which is collinear. Then the most likely explanation is that there is no a stop here, but there is an occlusion, which is quite common, widespread in the natural world. So one interesting thing is, can we prove that? Can we observe the same perception of partly occluded objects even in non-human species? And if so, could we observe that from the start in the absence of any experience? This can be investigated with young chicks, taking advantage of a phenomenon I will mention even later on, which is the imprinting. You know, the imprinting is the learning process by which the animals of some species, typically precocial species like chicks, learn to recognize an object by simply being exposed to it for a very short period of time soon after hatching. Usually is the mother hen, the chicken, but it works quite well. You know, obviously, the classical King Solomon's ring by Konrad Lorenz, even with artificial objects. So, as a tribute to Albert Michot, we imprinted chicks on a red triangle in this case, and then we presented the chicks with different versions of this bizarre mother. That is, a version in which the mother is partly occluded by a bar, and a version in which there is the bar, so same amount of physical stimulation, but the triangle is amputated, is lacking a part. So you can say it's amputated, or maybe there is a small triangle plus this trapezoid shape. You can describe it in several ways. This is the actual presentation of the stimuli. And what we found, yes, indeed, the chicks do prefer the partly occluded mother. That is, they behave in a way similar to the behavior of an adult of our species. developmental psychologists, my colleague working with human infants, were a little bit puzzling by our results. They are old results of some years ago. And they have developed a different procedure to investigate the problem with human infants, which is called habituation-dishabituation. So the idea is that they show the infants with a rod moving back and forth, partly occluded by this object. And then, after some time, after the poor infant is really fed up of this stimulus, they are changing it by removing the occluder and showing either the complete version of the rod or the two actually visible part, and therefore an amputated version, like in the example I showed you before of Stan Laurel and Oliver Hardy movie. So the idea is that if you perceive amodal completion, then you would be surprised looking at this stimulus, but not looking at this stimulus, because this is what you would expect if you have completion. Well, the interesting finding is that it took about 4 months to human infants to show evidence for being surprised when presented to this. The experiment was duplicated with chicks by a group of colleagues in England using exactly this procedure. So chicks were imprinted either with a complete bar or with a partly occluded or with the two pieces, and then they tested for the preferences between the complete and the two pieces. And you can see that, as expected, when imprinted on the complete, they prefer the complete, and when imprinted on the two pieces, they prefer the two pieces. But in the critical condition, in spite of the fact that the amount of red is exactly the same as in this case, they prefer the complete. That is, they behave as if, in this case, they would perceive a complete bar, which is by accident partly occluded by a black one. But there is also an interesting part of this story, which is puzzling, because if human infants would require 4 months in order to show amodal completion, this would suggest that in our species, amodal completion, recognition of partly occluded objects, would be the outcome of a learning process, whereas apparently in chicks this is available at birth, soon after hatching, which is puzzling in a sense, because it is probably consistent with our biases about the fact that our behavior is more dependent on learning than on innate mechanisms. But the point is that if we have available a mechanism, an innate, predisposed biological mechanism to recognize partly occluded objects, then why we should take the effort of doing that by learning? Learning is expensive in terms of the use of the nervous system tissue, and also is open to the possibility of mistakes. So the problem has been solved very recently by, and that's the moral part of the story, by a colleague in Padua who realized that human newborns have a problem in following with the gaze the movement of continuous moving objects because of the peculiarity of the development of our visual system. But they are quite good if, rather than using continuous motion, you simulate continuous motion with stroboscopic movement. I don't know whether you are familiar with stroboscopic movement. You are, in fact, because it is the basis of the movement that we see in the television, in the movie, in the cinema. It has been discovered by a German psychologist, Max Wertheimer, last century. And the experiment is very simple. He has 2 flashing lights. There was on of a flashing light and then off. And another one close, on and off. If the time interval is long enough, you see what is there. That is 1 flashing light and 1 flashing light. But if the time interval is short enough, you will see just one object that seems to move from one position and to the other. And this is obviously the basis, the mechanism by which we perceive movement in the the cinema, in the television, and so on. It's so-called apparent movement or stroboscopic movement. So what this colleague found is that if human newborns are tested using stroboscopic movement, they show evidence for amodal completion, for recognition of partly occluded objects at birth, just a few hours after birth. So the moral of the story is very simple, that even if something is not immediately available to an organism soon after hatching or at birth in a mammalian species, this is not evidence that it is not innate. A lot of mechanisms are innate, predisposed biologically, but they are not apparent immediately because maybe they require the development of other processes. So the mechanism for amodal completion is there, even in our species, at birth, immediately is predisposed by biological mechanisms, but it is not apparent until some other mechanisms for perception are developing in our species. We know this story because think about the problem of the walking of human infants. Sometimes we say children learn to walk. No, children don't learn to walk. They know how to walk. And in fact, if you take a neonate in this position and with the foot on a table, you will see the walking movement of a newborn. I would not recommend that you persist on that because the bones are not well formed yet, and there could be the risk of damaging the bones. But the structure, the mechanism is already available, and it is just the matter of waiting about 1 year of life in order that it would be apparent in behavior. This story of amodal completion is just an example, and there are several, of abilities that are traditionally interpreted or attributed to learning and to experience that could be, surprisingly, instead be innately predisposed in the brain. Another nice example is this one. You know this probably. It is so-called impossible objects. They have been discovered by Roger Penrose and his father, Michael. The father was a psychiatrist, and Roger was the physicist that was awarded the Nobel Prize a few years ago. And if you look at this image in a distract way, you could have the impression that it's a usual triangle in three-dimensional structure, but if you try to follow the structure, you will see that this object cannot exist in the real world. Actually, it can be built up. This has been done by Richard Gregory with a little trick
that is impossible, although it actually exists, made of 3 pieces of wood, just as impossible as the picture. Now, there's a space here which exists in the model, but not in your brain. I'm going to put my hand into that space. If I move my hand like that, it looks pretty jolly odd, doesn't it? Because that space is simply not in how you see this thing. I'll do it again, like that. So let's see how this is made. I'm going to rotate it, and you'll see that it's not really a triangle at all. It's an object like that, spread out and back.
So with a little trick you can build up the impossible object, but obviously it requires to be observed from a particular vantage point. So we did some experiment with these impossible objects like this, and again in this case we imprinted chicks out. It is obviously observing classical art like that of Mauritius Escher. You can see here an example. So we have a cube in which the critical junctions were covered by black discs, and chicks were imprinted on this object. And then these occluders were removed, revealing either a canonical, physically possible object or a physically impossible one. And we asked chicks to choose between the two mothers. And what we found is that, yes, indeed, they preferred the possible one. and note that they didn't have any experience of objects before the experiment. So what is going on here? Well, the experiment suggests that what is crucial is the way in which boundaries meet in images to form junctions. And this is a phenomenon which is very well known by visual artists. So T-junctions like this would suggest occlusion, whereas epsilon junction like this would suggest the presence of a three-dimensional object or a taxellation on the plane, as in this case. And this is a sort of alphabet which is already pre-established into the brain of vertebrates, both chicks, but also, I will mention this in a minute, human newborns. Let's consider more the story of the imprinting that I mentioned before. In the traditional view, the idea was that any kind of objects would be identically effective in order to obtain imprinting. This is a classical movie of Konrad Lorenz with the following response of his birds. But in fact, what we found recently is that if there are no alternative available, chicks or ducklings would imprint on any kind of artificial object, but they do have preferences. They have preferences which are related to all those signals, cues, that could be considered signals and cues of animacy, that is, of the presence of a living creatures. And there are several of these cues. Some are associated with the movement. For instance, a biological motion like this, you can recognize, I guess, or a dinosaur or a walking chicken, or even a simple one like, for instance, self-propulsion. This was suggested by Aristotle, that what is characteristic of animated objects is that they are capable of self-propulsion, whereas physical objects, inert objects, cannot move by themselves. And also there are stationary cues, such as faces or face-like appearance. Chicks, monkeys, even tortoises all show a preference for a very simple stimulus like this one, in which three high-contrast blobs are located in the shape of an inverted triangle, the eyes and the beak or the mouth, rather than the identical stimulus which is simply upside down. And for instance, we studied human newborns in my lab, and we presented human newborns a few hours old with the canonical face or the inverted, the upside down version, or this Picasso's like, that was the nickname in the lab for this version of the stimulus. And we use a particular technique which is not important to explain the detail, but basically the stimulus was presented at a certain frequency in order to measure the power spectrum in the EEG at that particular frequency. You can see here an example of the experiment. And what we found is that just when they were on average 40 hours old, so very, very young, there is a striking response to the face-like, to the canonical face-like stimulus with respect to the inverted and the Picasso's one. You can say, well, are you sure? I mean, you cannot prevent a human newborns of some degree of experience with faces, even very short. Well, no, we cannot be sure with humans, and this is the reason why biologists use animal models. With chicks, we can check for that. We can rear and have newly hatched chicks that we are completely sure they haven't seen any kind of face, and we presented different kind of stimuli, and this time, to be more precise, rather than measuring EEG, that is massive responses of the brain, we look for responses at the level of single neurons. So with very subtle microelectrodes, we measure responses of single neurons in a certain part of the brain, and what we found is shown here. You can also hear what you call the sound of thought, that is the spikes of the neurons. you can see that there is an increase in these spikes when the face-like stimulus is appearing, but there is very little response when there is just the beak. Again, very strong response. The line, not very strong. Very strong with the face. Not so strong with the Picasso slide. Very strong with the face. No response at all with the eyes. That was a surprise. So these neurons are there from the start. This animal has not had any kind of experience. It is recorded soon after hatching and reared for a few hours in condition of complete darkness. But face cells are already prepared in the brain. What is the function of these cells? Well, they are similar to the classical supernormal stimulus described by ethologists. They serve to direct the attention of the young animals to those features in the world which are more likely to be living creatures, and in particular social partners, the mother. Obviously, this sort of emoticon, they are not really similar to the natural faces, but this is not their function. The function is simply to attract the attention of the young creature to something which is similar to a face in order to learn about face. So these innate mechanisms are there, in a sense, in order to promote learning processes, or better, to make learning processes to proceed quickly. They are also the basis of the phenomenon of pareidolia, you know very well. The fact that we tend to see faces in the cloud, in natural and physical objects. This is the classical demon from the Twin Towers in the tree. This is my favorite, obviously, the famous Church of the Stunned Chicken. So what I am claiming for is a sort of Kantian message which is provided by the young chicks and the kind of research I am describing. Let's consider another example, which is numerosity. We did a lot of work, but also several other colleagues all around the world in different species, showing that animals are able to discriminate numerosity and also to count. This is an example. The chick is exposed to a different number of disappearing mothers, imprinting object, 3 on one side and 2 on the other side, and then after that he has to choose one or other of the 2 opaque screen, think a little, and then, you know, mathematics is not so easy. Obviously, we should be pretty sure that this is really respond to number and not to the continuous physical variables that co-vary with numerosity, like area, contour length, volume, and so on. So a lot of control experiments are needed. But the overall evidence is that there is indeed a mechanism to deal with number, which even in this case is encoded at the level of single neurons in the brain. You can see here an example. This is a neuron, a single neuron, there are several of them, that respond to number 5. So you see that every time that there is 5 dots on the screen, you see that there is a burst of activity, an increase in the activity. When the animal is presented with a single dot, which has the same area, in some cases with the same contour length, and obviously there are other conditions I'm not showing you, then there is not the same response. What is the basis of this mechanism? Well, this is a little bit technical, but I think you can follow me. If you look at the tuning curves of this neuron, you can see that the mechanism is essentially a mechanism of approximate number system. So look at the response of neurons responding to number 2. This is a neuron that would respond when there are 2 dots, irrespective of size, density, contour length, area, and so on. It has a peak of response for 2, but not only for 2. It is not an all or none. It is an approximate mechanism because there is a little bit of response, lower for 1, and a little bit for 3, and then lower again for 4 and for 5. And the same is true for all other these neurons. So they are filters that they have a peak of response to a particular numerosity, and that they respond in a gradual way to the other which are closer. And this is the basis of the so-called approximate number system. Even in this case, in chicks, These are animals that have had no experience with number and with numerosity, which suggests that number neurons are there in the brain, predisposed and prepared. If Franziska, I have another 5 minutes, but if I'm late, I would like to discuss another and final example that could be interesting with respect to the role and importance of animal research, even to change our opinion on human behavior. So this is a task we developed years ago on ordinal learning. That is, you know, number have a cardinal aspect, but also an ordinal aspect. So one thing is to recognize 4 as a cardinal item, but also you have to recognize that 4 is the number after 3 and before 5. So in this case, the chick is trained to select the 4th container, the 5th container from the left, or if you prefer, the 3rd from the right. And this is in an advanced stage of learning. And I'm showing you this video because there is something strange that probably you notice. Namely that the behavior of the animal is very stereotyped. It moved to the left and then ran along the overall series of containers. And you can say, well, it could be stereotype behavior by just one animal. But in fact, when we look at the recording of this behavior, we found that most, not all, but most chicks do the same. They move from the left to the right. And I remember at the time there was a visiting in our lab, a very eminent Australian scientist, that by joking said, well, Giorgio, look, they are counting from left to right, exactly as we do. And he was referring to a phenomenon very well known that probably you also know, which has been described by Francis Galton, the Darwin's cousin. And Francis Galton was the first to ask people to draw, to provide drawings of how they see numbers. And if you ask people, you could obtain very different and idiosyncratic example of the so-called mental number line. You can see here some example. But there is a regularity, not identical for all, again, a majority. And the regularity is that small numbers are to the left and large numbers are to the right. And you know that the traditional explanation for that is cultural and social. That is, it is the result, the byproduct of the reading and writing habits. In Western society, we have an habit to write from the left to the right and to read and to scan the images, and this could produce the biases. But obviously, the point is that even if I am championing the cognitive abilities of chicks, I must admit, reluctantly, that they cannot read and write. So that could be not the explanation. So we try to know that there is evidence in cognitive psychology that in society and culture in which there are different habits of reading and writing, Hebrew, Palestinians, for instance, just to take 2 examples at random, there is indeed a reduction of the bias, or in some cases even an inversion. So we did some experiment with chicks trying to reproduce this phenomenon. Very simple one is this one. We presented chicks with a panel with a certain number of dots, and behind the panel there is a reward, a wonderful small worm that chicks appreciate very much. And they learn the task very easily. And after that, we presented 2 panels this time with a number which was lower or higher with respect to the previous one. So 5 here, 2 or 8. And then we look simply at the direction of the choice of chicks. You can see here an example. So this is training with 5. No biases is observed with respect to the direction of the tour, to the right or to the left. It's random at this stage. This is a bit lazy chick, but it's coming. Okay, this is training. And that's test. With 2, they go to the left. But with 8, which is large, they go to the right. And this is a single chick, obviously we do that with a larger number. Obviously, small or large is a relative judgment, it's not an absolute one. So imagine that rather than training on 5, we have training on 20, very large. This case, 8 is small, which was large before. So what happens in this case? Let's see, training on 20 dots, and then after training, we presented chicks with eight, and this time they go to the left, because 8 is smaller this time, or 32, and this time they go to the right. And this is the data of a series of experiments we performed. So apparently there seems to be a similar phenomenon, and this was a surprise to my colleagues. They don't believe, to be very honest. So that was duplicated first in human newborns, and more recently by a colleague in France in honeybees that show exactly the same phenomenon. So maybe there is a biological basis for the orientation of the mental number line, but there was still, I have no time to discuss the mechanism, but I would like to discuss just this point. Studies of anthropology and ethnological colleagues seems to suggest that traditional society that have no any experience with numbers, and they have just an oral tradition, no writing and so on, they seem not to show this phenomenon, which is bizarre, because on one side we have evidence that human newborns show the mental number line with small oriented to the left and large to the right, and we have evidence in other animal species. Why then anthropological evidence would favor a cultural interpretation? So we decided to transform ourselves in anthropologists recently, and we compare, this time, Italian adults and preschooler with the Himba, which is a traditional society of Africa with only an oral culture. But this time, we use two different kinds of tests. One is a test similar to the one used previously by our colleagues with the Tsimane in Amazonia. This is an explicit task in which there is a series of discs with dots in it, and we ask the person to order, as they like, the disc with the numbers. And you can see here an example. And what we found, again, this is a bit technical, but I can explain simple words, is that basically only Italian adults show the classical bias with small number to the left and large number to the right. But the Himba, we have 2 field trips in 2021 and 22, they show a bimodal distribution. That is, and the same is also true for Italian preschooler before they attend school. There's 1/2 organized number dots, in this case, from right to left, and 1/2 from left to right. Then at this point, we decided to use a task more similar to the task typically used with animals and even with human infants, a sort of implicit task. So we presented on a screen a certain number of dots, and then a dot smaller in size in a congruent or incongruent position. That is, if small number should be to the left, then should be quicker to recognize than this one, and vice versa when there is more. And when we tested, you can see here preschoolers in Rovereto doing the test, and you can see here a warrior of the Himba tribe to do the same task. This time we found that all population, both the Himba, both the Italian adults and the Italian preschooler, do exactly the same. That is, they show the bias, small number to the left and large number to the right. And I think this is interesting because an observation which has been done originally as a test in non-human animals, chicks, and producing a bizarre result, something that seems to be very weird. Why chicks should have an oriented mental number line, has forced us to reconsider all the story about the mechanisms underlying mental number line. Now I am quite convinced that, sure, the mental number line can be modulated, is affected by culture, society, mechanism, and so on. No doubt about that. But the basic mechanism, the causal mechanism, is not culture or society, it's biology. And in fact, it could be observed in non-human animals, and it could be observed even in human newborns well before they have any kind of formal experience. You can say, well, is all this stuff just for basic science, or there could be some useful byproduct on that? Well, just to mention one thing, which was sort of serendipitous finding. After the work we did with the predisposition for animacy detection that I mentioned before, recognizing faces, biological motion, and so on, we reasoned that some kind of neurodevelopmental disorders, like for instance autism spectrum disorders, could be associated with some sort of deficit or difficulties, or for whatever reason, genetics or other, in these animacy detector mechanisms. You know, autism could be diagnosed only at about 2-3 years of age, which is eternity in terms of the experience. But there is a little trick that we can use, again, that is to study infants, newborns, which are so-called high risk for autism. That is, individuals that are siblings of other individuals that have been already diagnosed as autistic. So there is a couple that has had a son or a daughter, typically a male, with autism. They decide to have another one. They know that there is a risk for that. And there is a network in Italy, as well as in all other countries in Europe, that alert scientists of this high-risk autism individual, and we come with a mobile lab to go and test this when they are newborns, not at 3 years of age. And we use exactly the same tasks, the same test we develop for our chicks, slightly different because they cannot move, so we use fixation time, and they look to biological or not biological motion, and we measure their preferences, and we look for face and no face, and we measure preferences. Other tests I have no time to mention here, the direction of gaze, which is very important, that is if there is a direct or an averted gaze in a face. And what we found basically is that low-risk neonates, newborns, that is normotypical individual, they show the expected preferences. That is, they prefer the faces to the non-faces, they prefer the biological motion to the non-biological motion, and they prefer the biological motion to the rigid motion, etc., etc. But the individuals with high risk, they didn't show any preference. In some cases, actually, they show a slight preferences for the rigid motion, for the non-biological version, and the non-animacy version of the task. You can say, well, maybe they completely lack of the mechanisms for recognition of animacy, but the story could be slightly different, even more interesting. We have the first data of a follow-up at 4 months of age, and we tested these individuals at 4 months, and we found that, that's interesting, that typically development human newborns have the disappearance of the detectors because they are no longer necessary. Because at the start you have to look at human face-like stimuli, but then you are exposed to stimuli and then you learn about actual faces. But the individual of high-risk autism, this one, they tend to show at this point, at 4 months, a slight preferences. You can see in some cases it is also significant, for instance, for biological motion. So it seems that the mechanism for recognition of animacy is simply delayed for some biological reason. But the outcome of this is quite of a disaster, because obviously at the time in which this young creature would develop in ordinary condition the kind of mute dialogue of seeing, of gaze between the mother and the young is lacking, because this young creatures are not looking at the face of the mother, they are not attending to the movement, and they do that later on. So thank you very much for your attention. I stop with showing you a little bit of art. This is by Lucia Efferman, which is an American artist producing this beautiful movie. So thank you very much. If you don't manage to read Italian, I forgot to mention that the books are also available in English, not in Germany, unfortunately. Thank you very much for your attention. And Franziska, sorry, I have been too late, I know.
I would like to start because, Giorgio, the original idea why we invited you here is now we heard a lot about your methodology on how to detect biological determination of the brain programming. I heard you also discuss a lot about preferences that are interspecies similar on something like beauty, on something like proportions. And this is the realm which our institution, my discipline, is actually quite familiar with. So may I ask you to give us the main clues of what I heard you discuss in other interdisciplinary discourses, which relates very much with the methodological analysis, but maybe we go 15 steps further to discuss whether there are similarities that your research has detected.
Well, with respect to the mechanism I discuss, particularly the ones for animacy detection, that is the mechanism by which young animals recognize that something is alive, this seems to be really universal. Obviously, it is much more easy to study this in a precocial species like chicks for exactly the reason I mentioned before, because we cannot be sure that human newborns has being prevented by the adequate stimulation, for instance, seeing a face soon after birth. And as you can imagine, the mother of our human newborns in Rovereto Hospital would be not happy to allow us to do the perfect Kaspar Hauser, as the ethologists say, experiment, isolation experiment, would be not ethical, of course. This is the reason why we use animal models. But the kind of phenomena I describe, all have been documented in other species and as far as possible, even in human newborns. With respect to the question you mentioned, namely the recognition of beauty, that's very interesting. I think that we are, in a sense, in a right place, a right country to discuss about that, because the founder of the psychophysics, Gustav Fechner, was one of the first to investigate the psychological and, at the time, obviously not strictly neurobiological basis for the experience of beauty. I think that there is also a Max Planck Institute here in Germany which is called Experimental Aesthetic. Not sure it's in Frankfurt or in some other. Is here? Okay. So I guess largely inspired by Fechner. And Fechner did beautiful experiment concerning the golden section that you know very well, in which he presented different kind of rectangles to people, and he asked, tell me what is the best of the rectangle, the more beautiful rectangle. And what if, and he changed the size of the longer and the shorter side, and he found that apparently the beautiful one was that in which the ratio between the two sides was more or less corresponding to the golden section. That is the irrational number which is produced by the so-called Fibonacci series, in which you part with a number, we start with a number, and the subsequent is simply the sum, the addition of the previous one. So let's say 2, 3, and then 5, and then 5 plus 3, 8, and then 8 plus 5, 12, and so on and so on. And if you look at the ratio between successive number, you approximate an irrational which is called golden section, sezione aurea. It has been largely used by architects in the Renaissance. I am from Vicenza, my family, by the way, so I have familiarity with all the villas that Palladio built up using this kind of rules. But more generally, in terms of the biological mechanism, what could underline the experience of beauty, I think that we should consider seriously the possibility that sexual selection has been the basis for the development of disability in human species as well as in the other species. You know, sexual selection was the mechanism hypothesized by Charles Darwin to accompany or to explain natural selection, because he was very much concerned with the problem of so-called ornaments. That is, the kind of exaggerated traits, phenotypes that you could observe in animals in some cases, like the tail of the peacock, the horn of some deer or other animals. They are typically exhibited by males, and they are a problem from the biological point of view, because the animals that is taking this kind of ornaments is at risk of being predated or being unable to move with quickened velocity. And he has this fantastic, wonderful idea, Darwin, namely that the mechanism could be something like that. Let's imagine that at the start, a particular phenotypic trait, say the length of a tail, is a signal, a honest signal of how good is a male from the genetic point of view. That is, I have a good tail, and this means that I have in good health, for instance, or that I am so able to run that predators have been not able to catch me or to disrupt in part my tail. So at this stage, females would select this trait as a honest signal. The result, obviously, is that there will be a tendency in the population to select and to prefer by female those males that have longer and longer tails. You can say, well, the mechanism is not stopping when the signal is exaggerated with respect to the original honest function. Well, the answer is that there is a point in which the trait is literally shaped by the sexual preference of female. That is, females select males with the longer tail, not because it's useful, not because it's a signal of the goodness of the male, but simply because it is sexy. And if you select and if you choose to mate with a male which is sexy, then you will have a progeny, male progeny, that will be sexy. And this is the reason why these extraordinary forms, which are beautiful indeed, develop in the animal behavior, in the animal morphology. You can say this is nothing to do with the human condition, in which arts, beauty has mostly to do with culture, society, and so on. Yes, but consider the possibility, which has been suggested by some colleagues, for instance, that at the end, what is special about our species? The fact that we do art, poetry, theater, all this stuff. And imagine that this could be the neurological equivalent of the peacock tail, literally. That is, that they have developed initially as a way, I would say, to make impression, to convince female. This is, by the way, something that speakers, people that do talks, know very well. Sorry, noisy. Because it seems to suggest a major role of male, but no, because the selection is made by female, so you need a large and very impressive brains in order to judge the exhibition of beauty by males, ornaments or all the other tricks that males make in order to convince sexual partners.
You spoke about Konrad Lorenz. And Konrad Lorenz thought that imprinting was linked to a sensitive period, windows of plasticity in the nervous system. Last investigation has found that these periods are not rigid. In recent years, the possibility of reopening sensitive periods to allow for the possibility of nervous system plasticity is the focus of research in many laboratories.
Yeah.
Even in human, there are various sensitive periods associated with the development of language, stereoscopic vision, auditory perception, and so on. This prospect of making brain younger is very tempting.
Yes.
But it opens up ethical problems. Do you as a scientist think that in the next decade, this field of research could find clinical application or help to care for people with certain particular disease, or is a very dangerous double use?
No, I tend to have, in general, a positive and optimistic attitude with respect to science development. The point is that knowledge is good in any case. That's a Socratic idea. Obviously, we should be careful, and you should be in any way attentive, and we need to ask scientists to go out from the labs and to tell explicitly in a transparent way what they do, exactly what they do. That is the kind of experiments they do. For instance, I didn't mention, but obviously as a scientist that work with animals, I know I know very well that this is a sensible issue for a lot of people. And the strategy I use in my lab, I think even in other labs here in Germany, is the strategy of transparency. That is, everyone that wants to come to my lab and to see what we do and why we do, even very invasive procedure like the one I described today, recording from single neurons in the brain of a chick, I'm available to show and to explain why we do what we do. Obviously, at the end, we can remain in a disagreement, but in any case, that should be the correct way to behave, I think. With respect to the story of the critical period and the possibility to reopening critical period, yes, of course. You know that among the examples of phenomena which are associated in humans with critical period, obviously language is very well known, and also phenomena associated with the use of the eye. But another one is absolute pitch, that is it's called orecchio assoluto, the ability of some people to judge a note in the absence of other notes, which is rare, typically in musician. And there is some, and this is associated with a critical period, but there is some evidence that a substance, a drug, we are using it with chicks for different tasks, valproic acid, could reopen the critical period, and people treated with this valproic acid seems to show in adulthood an increase in the ability to make absolute pitch judgments. In the animal models, we can already be able to do this. For instance, in chicks, we show that using valproic acid and other hormone thyroid substances, we can reopen the period for imprinting, and also, interestingly, reopen the critical period for the kind of animacy detection that I described before. For instance, the sensitivity, by chicks at least, for the movement of the broody hen, or for the face-like stimulus, is typically restricted to a short period soon after hatching. But administering these drugs, we are being able to extend the period of this critical period. The issue here is not simply about the ethics, it is also practical one, because obviously if you reopen a critical period, you make the brain plastic, similar to the condition of a young creature, of a human newborns. And this means also the risk of losing, because of the mechanism of reconsolidation, the memory which has been already fixed in the brain. I don't want to discuss too much about that, because it's the topic, the trick that I use for a novel I have just completed, and that will be published, I hope, in September. It's my first attempt to move in a different realm, that of novels, of the literature, and I am discussing exactly this kind of phenomena together with several others.
Danke, mille grazie. First slide. Okay,
yeah.
Thank you very much for your insights. I was just thinking when we were talking, yes, we're reading from left to right all the time, but Arabic on the other side is from right to left. So, but this was just as my real question, and maybe it's too big to be answered, but I never had the time to bother about this subject, but it's just starting to interest me. It's about intelligence. So how is intelligence, what it is? It's a huge question already, but how is it generated? So what is like genetic? I just read once that in the first 3 years of our lives, intelligence is like to be defined, and then it's like over. I don't know. It's just, as I said, I never had the time. Can you contribute anything, or the question is too big?
Well, my suggestion is that it is probably premature, but it would be good in the near future to avoid using general terms like intelligence or language. For language, we have exactly the same problem. From time to time, there is the discussion, is language unique of our species? Well, language is such a big thing that includes syntax, semantics, pragmatics, phonological aspects, and either one of these specific computations that are done in the brain can be discussed and examined separately. So, for instance, there is good evidence that some aspects of language are shared with other creatures, but for instance, apparently, syntax seems to be quite unique of our species on the basis of the current state of knowledge. Same, I think, could be, the same attitude should be maintained for intelligence, because it's a very large term, which is used in ordinary language, but from the scientific point of view, it reflects very different abilities. And this is also the correct way to consider intelligence from the biological point of view. What is intelligence in what is the most general, large definition? Well, intelligence is the capability to solve problems. But the point is that the environment asks different kind of offer different kind of problems to biological organisms, very different. So the problem of recognizing a face is different from the problem of orienting in space. The problem of computing quantities and to do arithmetic operation is different from the problem of recognizing an enemy or deciding whether to cooperate or not to cooperate with other members of your species. Each one of these are forms of intelligence. And given that different sources of problems are available to the biological organism, and different kind of algorithms and computation have been developed in order to solve specific problems, we can imagine that different mechanisms are there in the brain. And we know that very well. The mechanisms for face recognition are localized and are quite different from the mechanism associated with with respect to the logic, transitive inference, for instance. It could be that there are forms of intelligence in which you could observe something general, because the environment asks for general question in all ecological niche to organisms. For instance, the ability to develop an understanding of causal relationship. And causal relationship is mainly managed by mechanism of associative learning, Pavlovian and instrumental conditioning. And this seems to be universal. From planaria to Einstein, associative mechanism seems to be quite similar. But other kind of intelligence seems to be highly specialized. Consider spatial memory. There are food storing birds, very well known, which are able to food caches in a natural environment at the end of the summer in a number which is astonishing. That is, they are able to have thousands of food caching, and they are able to recover month after the position of these food caches in a natural environment. And they do that on the basis of spatial memory. This is on allocentric representation of space. Now, consider the case of human beings. How many food caches you would be able to remember after a couple of months? The estimate is that we would be able probably to remember 9 items. So no story, no comparison with the food storing birds. They are apparently much more intelligent in this particular kind of task. But obviously, every species has its own adaptive specialization. Consider a little trick. I can take paper and a pencil, and I go in the natural environment and I prepare a map. And in this map, I select, I indicate in a very precise way the different position of the food caches. And the only limitation on that is how boring is the activity. After that, I can take the map, put into a drawer, and wait not just a couple of months. I can wait years. And if the environment has not been changed, then I go and make the race with the food storing birds, and I will be the winner. But obviously you can say, this is not very fair, because the food catching birds is using just a little piece of the brain, the hippocampus, where they have this extraordinary spatial memory, and you are using something different, a map that is symbolic thinking. But this is exactly the point. That is, exactly as the food storing birds has its own specialization, in which is super clever, spatial memory, we, as humans, we have our own specialization, symbolic thinking and language, for instance, which is unique of our species and is not shared with food storing birds.